This video is going to show you how to set the oocytes up for injections. First, we're going to go get the pen strap solution that you will use to keep the oocytes in. That's in the back. Go through this door. And it's this one with the green handle. You open it up, and here on the top shelf, we have a bottle of pen strap. That's what you want to use. Every 2 weeks, you need to make a new solution of pen strap, because after 2 weeks or so, this solution gets a little old, and it's better just to make a new one to keep the oocytes healthy. We'll grab the pen strap, and then also in this cabinet, we're going to grab this little dish. It has, if you look at it closely, you'll see it has a grid in the bottom, and that's to hold the oocytes in place so that injections are easier. This is what we place them in. Come and you put the tray on this little platform. You put in just enough pen strap to cover the bottom. You don't need a lot. Just like that. And then next you use a micropipette that has a larger tip. We like to use this one. It goes up to 200, 20 to 200. And use this tip. Sometimes you have to clip the tip off a little bit if you need a bigger opening for the oocytes. And you'll use this to gently transport oocytes from their shipping container, this little vial, into here, so that then you can inject them. How to pull more tips for injections. This is the machine that you will use for pulling those tips. The on-off switch is here on the side, so we're going to turn it on. Over here you have labeled what program you have to use for injections or impaling. We're going to pull injection tips. So we come over here. It says, what do you want? Here's the number pad. We put in 65 and push enter. And you can see that the correct program pulls up. We'll open the top. For injections, the tips that you will, or the tubes that you use to pull tips with, are these ones. They usually have a green label on them, but here is the size that you want, and you can just shake them out of this little container like that. The whole key about pulling tips is you want to make sure that you do not damage the filament that is in this box. So to do that, you have to carefully put this tube in. So you put the glass tube in that track, that it slides along this track here. You loosen this a little bit, and you can slide it through. Once you get about to the edge of the track, I like to tighten it a little bit. Then right over here you can see there's a little lock. You have to push that in, and then you can slide this whole thing. So you have to push right here, then this will slide. So you slide this over. You also slide the other side over. It also has that same lock. Hold both of those, and then continue to push the tube. Make sure it stays in its track. I'm going to tighten that a little bit more. Push the tube until it's about equal on both sides. So there's just a little bit of overhang, maybe a little less than a centimeter on both sides here. Then you tighten down both sides until it's just snug, just so it stays. Close the lid. And then pull right here. It'll start to hum a little bit. Soon you'll see a glow as the filament heats up. and then that loud noise lets you know that it's done. You can open the lid and take out the new tips. So you just loosen this until you can pull this new tip straight out. As you can see, it's really fine and delicate, so you don't want to let that tip touch anything. And then you put it in this little case that we have just for holding the tips. Take the cover off. Just place it. Try to put the tip so that it's resting in the air. And that's how you pull a tip. After pulling a tip, but before using it on the injection machine, you first have to clip the tip to the correct diameter. So I'm going to show you how to do that now. The first thing you have to do is RNase out your hands. So here's a bottle of RNase out. You do this because the needle or the glass tip that you're using is going to be holding RNA, which is easily degraded. So you clean your hands. It's okay if it's still wet, it's fine, before you touch the needles. Now I have my needles here. Make sure that they are the injection needles and not the impaling needles. Take one, and I'm going to bring it over here to the rig. And here on the rig, you can see that I have the tray upside down, so I can just place my needle on that. And then I'm going to make sure I'm on the highest magnification, which is 12, and then clip the tip to a diameter of 2 ticks, which is about 2 micrometers. But on the highest magnification, the tick marks line up to about, well, you want to clip it to about 2 ticks wide. And I just use this to clip it, and then it's at the right diameter that you can use it on the injection machine. Now that you have clipped the tip of your injection needle, the next step is to prepare it and put it on the injection machine. So to do that, you take this sterile mineral oil, and you have to fill the needle with mineral oil, making sure not to leave any bubbles. So I like to go all the way down to the very tip and slowly inject some, and then just fill the needle as you pull it out until the entire glass tube is full of mineral oil. And see, I got a little bubble in there. There's still a little bubble in there, so I have to get that bubble out. So I'm just gonna go ahead and poke the needle back in underneath the bubble, and then put in some more oil until the bubble comes out the top. Okay, now this is ready. I'm just gonna dry it off. We're gonna take it over here, and we're gonna put it on this machine. So first, if you wanna look right here, you unscrew this little black tip, and inside are 3 pieces. Those 3 pieces come out when you put the tip into this way. And then you can push out those 3 little pieces. I'm going to lay them out here so you can see them. Black, white, and black again. They have different size holes in them. So the black piece with the smallest hole goes back on this little plunger on the machine. So you can put that on right away. Do that first. Just like, oh, come on. Just like that. Then make sure that your glass piece is all the way through. You can take the other black piece and it goes all the way around the glass tube, like that. And then you take the white piece, and the white piece has a hole on one side that is big and on one side that is small. The big side goes on to top of the glass tube, and it just sits on top of that, just like that. And then the small, move that out of the way, the small piece fits around the plunger. And so all 3 go on, then you can push this piece up and screw it on. Then the last step that you need to do to prepare the needle before injections is to empty it. So if you come over here to the control panel, you just push and hold empty until it beeps at you again and the plunger is completely into the glass tube. Now that you have the oocytes prepared in the dish, clustered in the center so it's easy to keep track of them, and the needle is also prepared and ready to go, now our next step is to ready the RNA. The RNA is the last step because it degrades easily, so we want to keep it contained as much as possible and not exposed in the air. The first step is to wash your hands with the RNase out, just to make sure that you don't degrade the RNA with any contaminants. The next thing we need to do is to prepare our micropipetter. So we're going to use this one. We have 12 oocytes in our dish. This is the 0.1 to 2 micropipetter, and we have it set to 12. So 12 oocytes, 12 on the micropipetter, 1.2 microliters. And that will be the correct volume of RNA for us to use in a minute. Then we go and we're going to grab the lid that we're going to put the RNA in before we draw it up into the glass tip. So up here on the shelf we have a bunch of small centrifuge tubes, and you shake them down to the top before you open it. You don't want to reach your hand into the bag because we're trying to keep it as clean as possible. Seal it up again, put it back. Now the only part of this that you want is the top. So you just break the top off and you can throw the tube away. We're going to set our tube back in on top of that platform. And now we're going to go get the RNA. You want a bucket that we're going to fill with ice. When you're transporting the RNA, always transport it in ice so that it stays cool and doesn't get degraded by the temperature. And our RNA is stored in this fridge, or refrigerator, it's a freezer. It's in the door right here. This is our tray of RNA. And we have different sections labeled, as well as labels on the caps of all of the tubes. So today we're going to do alpha 7 beta 2. So I've prepared this aliquot of alpha 7 beta 2, and this is what we're going to use. So I'm going to take it out of the freezer, put it in the ice, and then take it back into our room. First thing you do is defrost it, so just hold it in your fingers for a few minutes, it doesn't even take a minute, and it'll defrost pretty quickly. Just keep track of it. There you go, it's already melted. You want to vortex it for just a minute to make sure that it's all suspended. Now, ours looks good, but sometimes if the RNA ends up in the cap, you'll have to centrifuge it back down to make sure that your droplet of RNA is in the bottom of the tube. Now we're going to go use this, put it back in the ice. You have your micropipetter. Here we have the micropipetter tips that are specifically for RNA injections. You want those the least exposed possible. Make sure the tip of your pipette doesn't touch anything. If it gets contaminated, then you have to redo it. Draw up the RNA. Place the cap, put it back in the ice. Dispense the RNA into this lid that we just got earlier. Make sure that you get all of it out of your micropipetter. And then before this RNA is exposed for too long, now we need to draw it up into our glass needle. So this knob moves your needle straight down.
Show the knob again.
This knob will move it up and down, like that, in this way. This one moves it side to side, and this one moves it forward and backward. Forward and backward. So between those things, and you can always move the platform itself. Position it so that the tip of the needle just barely enters into the liquid. You have to be careful not to break the tip of your needle by going down too far and pushing it against the platform. And then once you're in the liquid, you go back to your control box right here and you push fill. And you have to watch it to make sure that the liquids doesn't jump around and that your needle stays in the RNA suspension. And you draw up all the RNA, and then once that's done, you take this and put it back in the freezer.
Cell wall on the OS, not the cell, but the cell membrane.
Yeah.
It's just so thick.
It just kind of bounces off and like rolls around.
It just rolls around and you're just like chasing it the whole.
Oh, there we go. This is a good one. I thought.
One last tip that I forgot to mention is I find it a lot easier when you're doing the injections to move the entire tray up and down to get to the other oocytes rather than trying to move the needle every time. If you can keep your injection needle focused and in the center of the microscope, it's a lot easier just to move the whole base to the next oocyte. And now the last thing that you have to do is clean up. After all of the oocytes have been injected and you take them back and put them on that oscillating dish, the next thing you have to do is take care of all of this. First, you push fill, which will pull the plunger all the way back up. So you have to just push and hold fill until the plunger is completely retracted into the machine. Then you can pull off the glass needle and just throw it away in a sharps container. This dish needs to be taken to the sink and rinsed with DI water. Just rinse it quite well and then put it on a paper towel in the cupboard to dry for next week. After that, make sure you turn off the work lights and return the pen strap to its place in that incubator. That's what it actually is. And then you're good to go.